Next, we have our first oral presenters. This is one of the abstracts submitted. It's Bazgir from Genentech. Hand over to you for your talk.
Yes. Thank you for the introduction. My name is Bazgir. I'm working at Genentech as a machine learning scientist in the development sciences and Genentech Research Early Development Organization. I have the pleasure to represent Genentech today at this stage to present our work on integration of graph neural networks and Neural-ODEs for tumor dynamic prediction. So I would like to start first with what is tumor dynamic. So as you know, tumors are abnormal cells in the body that they divide and proliferate very quickly and cause into issues that we call them sometimes cancer. And it's very important to analyze and understand the tumor dynamic in different cancer types and oncology clinical trials, for instance, because we want to know how these tumors are basically growing, if the treatment is going to help in terms of like shrinking the tumor, or if the tumor is going to rebound after some time. And these are very important in phase 1 clinical trial. Phase 1 is basically when you are dealing with animals or preclinical stage, or when you are in phase 2 and phase 3 when you are dealing with clinical or human data, basically. And just as an example, these are just like some examples of like synthetic patient data, as you see, just giving you a flavor like what tumor dynamic look like. Typically, the samples are very, very sparse, and you mostly cannot get that many data longitudinally, because we follow up the patients in clinical trials longitudinally. We want to know how they are responding to a treatment. So what are the benefits of basically studying tumor dynamic and why we actually do this? There are many benefits here. One is basically in improving the treatment plan, like personalized therapy or drug selection and dosing optimization. So we want to know if a patient has this tumor at this time point and we are giving this drug with this dose, based on the model and the prediction that we get, how we are going to optimize the dose, basically, or how we are going to change the treatment, and so on. So it helps us in terms of patient selection even, do we even need to include this patient in a clinical trial or not? It helps us, endpoint prediction and resource allocation. Okay, given this, for instance, we have seen this pattern of the tumor dynamic in phase 1 clinical trial. And based on this tumor dynamic, do we want to, have this many patients for the phase 2 or phase 3 and so on? And also it helps us to understand how long we need to follow the patients, basically reducing the phase 1 trial duration and making go, no-go decision making. Basically, at the end of the day, are we going to file basically this drug or the package of this drug to FDA and get approval or not? So these are very, very critical, information that you can get from tumor dynamic prediction. But there are so many challenges here. One challenge is most of the high throughput and high quality information that you get, like genomics and so on, you cannot keep asking the patient that, hey, I want this genomics profile at this time. Typically we get them at the very beginning, right? So at the day 0, basically, before giving treatment to the patient, or mostly at the end. But there are some evaluation that we do along the line, or some measurements, some imaging data that we get to know what is the tumor size. So basically, you want to understand the pattern longitudinally, but most of the information you have are at the very, very beginning. So, and these informations are very heterogeneous. It could be from Genomics profile, or in genomics profile, we have different, level of information, the RNA, protein, and so on. So we wanted to come up at this study with a approach that we can integrate all this information into a model such that we can integrate heterogeneous information and also predict a longitudinal tumor volume along the line. So for that, we have multiple blocks here. One block is taking care of like heterogeneous graph encoder, which basically the job is taking care of representation of drug, disease, and genomics profile of patients. So basically, if I have a patient who has this disease and I'm giving it this drug, what embedding I can get from it. And we can represent that with graph and basically get that embedding very efficiently with graphs. On the other hand, when we do decision making, sometimes we want to know, okay, if I know X amount of days of information about the tumor volume of the patient, how I'm going to react afterwards, or how I'm going to change the treatment after that. So basically, you need a block that basically can give you embedding of the observed duration time. So because of that, we designed like a tumor encoder, which it basically takes the early tumor data and then put that into an embedding. And at the end of the day, these two embedding goes to as an input or the initial condition of a Neural-ODE system, where the Neural-ODE system with like an MLP block after that will help us to basically capture the trend of the tumor volume longitudinally. So for the graph, we have basically two graphs, like GCN and bipartite GCN. So GCN is mostly used for the genomics profile or RNA graph, RNA-seq. And bipartite graph was used for getting the information between like drug and gene, and also disease and gene. I'll go after that. I'll explain one by one. So for the RNA graph, we started with pre-training a gene-gene graph. We had a knowledge graph of like for multiple diseases, what are like basically the knowledge graph, how these genes are being studied and how they are related to each other. And typically these graphs are extremely large. So it's like a graph level task, a prediction task, and the graph like typically have up to 20,000, sometimes even 50,000 nodes. And each node represents one gene, and the edges or the link between the nodes are basically a connection between the genes in that disease, basically. So what we did, we initially pre-trained the graph using three different loss functions. One is node reconstruction, the other one was edge reconstruction, and the other one was KL divergence, like as a VGAE graph, and we use that as to pre-train the graph for downstream tasks. So once we have that graph for RNA or the genomics, now we want to relate it to disease and drug. So we know that for each patient, we already know the disease, and we know the drug that we are trying to give it, right? So we need to find the right embedding for these two, given the genomics profile that we have for the patient, right? So we have to first reconstruct or create the bipartite graph between drug and a gene. So what we do, we create like a bipartite graph through attention convolution that basically passes the message from drug to gene, and then from gene back to the drug to update the embedding of that drug. We do very similar thing for disease and gene as well. So we have another bipartite attention convolution layer, which basically gets the embedding from RNA or the gene graph and update the embedding of the disease. And at the end of the day, we have the embedding for the disease and for the drug. We concatenate them, and we call them beta 1. So, and then now we want to go to the other step, which is tumor volume encoder. So basically, if you look into this gray area, so it basically, we want to know, okay, if I have the information for this particular patient, for this, like in this gray area, like basically from day 0 to day 7, how can I predict from day 7 to the rest, right? Like how basically the tumor is going to respond to a treatment given the genomics profile I have from the patient. So what we do, we basically implement an RNN encoder to encode these early tumor data, or the observation window we call it, into a space, embedding a space, we call it beta 2. And then beta 1 from the graph encoder and beta 2 from the RNN encoder, we concatenate them and we use them as the input of our Neural-ODE system. But you may wonder why we use Neural-ODE. In oncology, there has been very established studies of like mathematical modeling of tumor dynamic, and most of them are based on ODE systems. For instance, this equation I have on top, it's from Zhu et al., recently published at CPT. So they are able to very good extent basically capture the trend in the tumor dynamic. They also have other benefits like counterfactual simulation. Counterfactual simulation are basically what-if simulation. Like basically if I have this drug with this dose, and what if I want to double the dose or half the dose? These are cases where ODE systems are very helpful. So, and also in terms of interpretation, you can interpret that what component goes back to basically growth or shrinkage or rebound and so on. So that's why we use Neural-ODE. So at the end of the day, we use beta 1 and beta 2 as the basically initial conditions of our Neural-ODE, and then we have another F, which is a neural network, to basically capture the pattern in the data. So we wanted to evaluate our system and see how does it work. We evaluated it based on a criteria called RESIST, or basically response evaluation criteria in solid tumors. It's a very well-known criteria in oncology and in clinic. So at the end of the day, what it does, it categorizes the patient response into 4 categories. One category is a complete response. It means that the patient responded to the treatment in the best possible way. Then we have partial response, we have a stable disease, and we have progressive disease. So it's basically a spectrum from having the best possible response to have no response at all. And we wanted to evaluate and see how our model perform on different observation window. Basically, if I observe the first 7 days of the patient and try to predict the rest, and if I observe the first 14 days and then I try to predict the rest, and so on. So we evaluated that by having the graph encoder and not having the graph encoder. And as you can see here in this plot, in all the metrics, having the graph encoder basically improves the predictive performance. And also, obviously, the more you observe, the better predictive performance you have, right? So we wanted to also evaluate that at individual level. Here are some individual level prediction for some patients where we compare our approach with TGI, which is a well-known traditional approach for tumor growth inhibition modeling. And as you see, like obviously I'm biased to my model, so I'm just showing you the best possible results. So here we have basically for 4 patients where we have patients where our model is able to predict the response properly with respect to the RESIST criteria. So for instance, on the top left, we have a patient which is progressive disease. Our model is able to capture that as progressive disease, but the TGI metrics is basically classify that as stable disease. And so on and so forth for the other patients. We have another patient with like complete response, which TGI consider it as partial response. We have another patient with stable disease, which TGI consider that as progressive disease, and so on. So at the end of the day, I wanted to basically acknowledge the contribution of my colleagues. They were very basically, their contribution was very significant to get this work done and publish it on ICLR. My colleagues are Zichen, Jiwon, Mark, and James. And if you are interested to our work, please go ahead and read our paper. It's publicly available. And the conclusion here is, with this framework, we are able to propose that how we can predict the tumor dynamic and integrate RNA-seq treatment and disease into a model along with early longitudinal tumor data to be able to predict tumor dynamic. And we have also shown that how it improves the Neural-ODE performance, and also how it compared it with TGI models. And for the future work, we are planning to work on interpretability and see if basically we can get basically given the tumor location and the drug target that we have, can we identify like the right gene that the drug is targeting. So that's it. If we do have time for Q&A, please.
That's great.
Thank you.
We've got time for maybe one question, but we're running a little bit behind. So if anybody has a quick one, you can take one question.
Thank you for the great talk. Can you tell us whether you ran ablation studies and whether of beta 1 or beta 2 you found that one was prevalent in terms of productivity?
So beta 2, which is, thank you for the question, that's a very good question. Beta 2 is like the most important one, which is based on the early tumor data. But this is something that is very well, could be included in the model by not even using like multimodal approaches. The benefit is using like the graph information, and that is something that previous models like has not incorporated them yet. So if I want to give them importance, I would say beta 2, which is from the early tumor data, and then beta 1 from the graph encoder.
All right. Let's thank Hamid again.